dear colleagues. Today, I am pleased to present our commentary published in iMeta, titled The Role of Human Microbiota in Breast Cancer Pathogenesis and Treatment, co-authored by me, Professors Yiding Chen, Xuli Meng, Xiaojia Wang, Manli, and collaborators. In this commentary, we summarize current advances in understanding the relationship between the human microbiota and breast cancer. We focus on the potential mechanisms by which breast, gut, and oral microbiota contribute to breast cancer development, as well as their impact on the treatment response. We also discuss microbiota-targeted strategies and their translational challenges, aiming to provide new insights for precision medicine and therapeutic interventions in breast cancer. Now, let us dive into this emerging field. First, let me introduce the background. Currently, breast cancer remains the most common malignancy among women worldwide. Treatment resistance and inter-individual variability in therapeutic response are major clinical challenges. For a long time, the breast was considered a sterile organ. However, recent studies have revealed that breast tissue actually harbors a low biomass microbiota, with significant compositional differences between healthy and malignant tissues, suggesting that local microorganisms may participate in tumor initiation and progression. Further research has demonstrated that, in addition to the local breast microbiota, the gut and oral microbiota also influence breast cancer biological behavior through local and systemic crosstalk, and may modulate treatment responses. Of particular note, accumulating evidence indicates that core breast cancer treatment modalities, including immunotherapy, chemotherapy, endocrine therapy, and HER2-targeted therapy, may all be regulated by the microbiota. Therefore, a deeper understanding of these complex interactions holds promise for discovering novel biomarkers, optimizing therapeutic strategies, and developing microbiota-targeted interventions, ultimately improving diagnosis, prognosis, and precision medicine for breast cancer. Now, let me transition to the key findings. Microbiota from multiple ecological niches, including the breast, gut, and oral cavity, participate in breast cancer development through local and systemic crosstalk. The microbial composition differs between tumor and normal breast tissues, involving phyla such as firmicutes and bacteroidetes. However, heterogeneity across studies remains. Specific bacteria, such as Fusobacterium nucleatum and Bacteroides fragilis, are associated with tumor initiation, proliferation, metastasis, and drug resistance. These microbes regulate key processes, including T-cell infiltration, inflammatory cytokine release, estrogen metabolism, and apoptosis, through immune, metabolic, and inflammatory pathways, thereby shaping the tumor microenvironment and breast cancer progression. Gut and intratumoral microbes can influence immunotherapy response in breast cancer by modulating immune cell infiltration and inflammatory cytokines. Microbiota and their metabolites affect chemotherapy efficacy through drug metabolism and apoptosis, while changes in microbial composition are also correlated with chemotoxicity. Moreover, microbiota regulate endocrine therapy sensitivity and resistance by mediating estrogen enterohepatic circulation and local reactivation. Microbiota-targeted interventions, such as probiotics, fecal microbiota transplantation, and engineered microbes, hold promise for improving the efficacy of various breast cancer treatments. However, clinical translation remains at an early stage. In summary, first, the human microbiota is closely associated with breast cancer biology, involving multiple ecological niches including the breast, gut, and oral cavity. Second, microbiota and their metabolites influence the tumor microenvironment and breast cancer development through multiple pathways, including immune, metabolic, and inflammatory pathways. Third, microbiota and their metabolites may correlate with responses to systemic therapies, including immunotherapy, HER2-targeted therapy, chemotherapy, and endocrine therapy. Fourth, microbiota-targeted interventions show potential to enhance treatment efficacy, but clinical translation is still in its early stages, and future efforts should strengthen mechanistic and translational research.